Happy evening everyone out there. I believe that you all stay safe and healthy. I am Dr. Joe Patrick Namraj, Professor in the Department of Mechanical Engineering. I would like to brief on our campus profile. Our institution, Francis Xavier Engineering College, fondly known and referred as FXEC, is located in Tirunelveli, the sixth largest city in the state of Tamil Nadu. Tirunelveli is the hub of many educational institutions and it is well known as the Oxford of South India. FXEC come under SCAD group of institutions and educational wings of SCAD, a socially committed group in Tamil Nadu. Our institution has been offered autonomous status in the year 2019. We offer 6 UG programs, 10 PG programs and have scholars carrying out their doctoral studies in our research center. The Department of Mechanical Engineering is the largest department on campus with around 600 plus students, 35 faculty members and good infrastructure in the terms of laboratories and other amenities. We offer BE in Mechanical Engineering and ME in Industrial Safety Engineering. The UG program is accredited by NBA, the National Board of Accreditation. Apart from courses defined in curriculum, we offer a number of value-added courses and impart various skill training to the students. In addition, well-furnished laboratories that come under curriculum. We have few special laboratories to encourage innovations. We also have strong academy for training students to appear for GATE exam. Thank you. I welcome you all for this session. I hope you will have a productive time. Thank you again. Thank you.
Hello everyone, this is Dr. Rizzi, Professor in the Department of Mechanical Engineering, Francis Xavier Engineering College, Tirunelveli. I'm here to brief on the profile of today's speaker, Dr. Gyojin Cho. Professor Gyojin Cho completed his BS in Chemistry in the year 1989 from the National Science College, Gyeonggi, South Korea. He completed his PhD in the year 1995 from the University of Oklahoma, Norman in the United States. He started his career as a teaching faculty in the year 1996 in the Department of Chemistry in Suncheon National University, South Korea. In 2008, he was appointed as the Professor of Printed Electronics Engineering, World-Class University Program in Suncheon National University, South Korea. In 2019, he was offered a prestigious position as a Professor of Biophysics in the Institute of Quantum Biophysics, Sungkyunwan University, South Korea. In addition to his regular teaching, he has shared his expertise as a visiting researcher and also holds some prestigious positions in various institutes and research laboratories. He was a visiting research scholar in Rice University, Houston, Texas, in the United States from 2004 to 6. From 2011 to 13, he was a visiting research engineer, and from 2013 to 16, he was a visiting research scholar of electrical engineering and computer science in the University of California, Berkeley, in the United States. In 2013, he was serving as a visiting professor of Materials and Construction Laboratory in Tokyo Institute of Technology, Japan. From 2005 to 2019, he was serving as a chair of Regional Innovation Center, Specialized Printed Electronics in Suncheon National University, South Korea. From 2007 to 12, he was acting as a chair of Research Consortium for developing roll-to-roll printed RFID tags. To look at his laurels, he has published many books, book chapters, and has also published a good number of research papers in reputed peer-reviewed journals. He holds 3 patents to his credit. During the last 10 years to lead printed electronics, he was a principal investigator of 2 government R&D projects worth $20 million for the US and developing technology for industry for 5 years from 2008 to 2013. He was serving as art tour session organizer in Materials Research Society, Boston from 2014 to 16. He is the Associate Secretary of International Standardization, IAC-TC 119, Printed Electronics. He also has served as Associate Editor in Flexible and Printed Electronics, IOP, on editorial board in Scientific Reports, Nature Publishing. In 2017, he demonstrated the fully auto or gravure printed TFT active matrix-based flexible e-paper for the first time in the world. So we have among us Dr. Gyojin Cho, one of the greatest researchers, innovators, and professors who is known across countries and have international credentials. I hope every one of you make use of this session effectively. Have a great time and thank you.
Okay, then. Hi, everyone. It's my great pleasure to see you through this internet lectures. I hope you enjoy my talk. Actually, which is regarding for large-scale additive manufacturing process, which abbreviates the LAMP for Biochip. Of course, since this is the only 30-minute class, I couldn't cover whole in detail, but I would like to show you and introduce you about the outline and why we are interested in the biochip and what is the goal and why we need a large-scale additive manufacturing process and then what it is. Okay, so that is what I focus on during these lectures. So I would like to start my lectures on this based on some two stories. Actually, the current medical service, as you can see it here, then, for example, in here, if you visit the China, which is, as you know, the coronavirus was invoked from there, and then you have to wait to 6 hours in hospital to confirm negative or positive result from coronavirus test. But if innovative medical service, you don't need to wait that long time. You just to get the in-situ output is a positive or negative result in less than 3 minutes at the airport. Okay, that is the one we are learning for the research to reach the innovative medical service, especially for diagnostics, for like infectious disease and this kind of thing. And also the other thing is we also consider that, for example, a man 60 years old confirmed the pancreatic cancer, 3 stage, and get chemotherapy for 6 months in a hospital, but he died by side effect because the chemotherapy, it can kill your cancer cell, but it also can kill the normal cells. So current medical service, if you are lucky enough, you may survive, but not, then you will die because they dose the drugs, anti-cancer drugs to everyone, same thing. But in the innovative medical service, it's like for your same situations, but you will get the chemical therapy with personalized anti-cancer drugs, which is selected by organoid chip. That is the only just $15. So it will be not very expensive. So, and then you will cure and then get the more than age 100. And the other target is, example is if you are working the pharmaceutical company and develop the new drug and run the clinical trials to approve FDA for 3 years, but finally they drop out the new drug due to the cost and they couldn't survive for the clinical test. However, if you're using our new developed the drug screen test kit, which is the kind of arrays of organoid, and then you will get the all clinical tests in a day, actually, with the very low cost and get the FDA approved. And so this like one example, I gave a typical 3 examples for our innovative medical service. So in our lab is focused on this kind of to develop the medical service and with the low cost. Okay, so I would like to show you for not exactly how detailed to develop the innovative way, but instead I would like to give you some brief ideas what we are doing it and why we are doing it, and then what kind of method, manufacturing method is required to reach there. So for the biosensors, we need to detect the biomarkers, whether in the low concentrations. For example, in our body, if you tracing back to at the end, it will be all ended by all molecules. Okay, so of course, if, for example, if you go to the brain, nervous, and then intercellular, intracellular, and then we'll go to the molecules. So we often test the cell, but finally we needed to separate out DNA and RNA and this kind of the molecules, even sometimes ions, and then needed to be detected. And what concentration we needed to do it. But if it is a normal millimole range, it is easy to detect. But in our normal life and fluid, it's like blood or urine and saliva. There is not much concentration of the DNA, RNA you can detect. It is usually the femtomole or attomole, very, very low concentration. So it is really hard to find your DNA copy from there. Okay, so and that kind of thing we needed to develop to avoid this kind of thing. We needed to develop very high sensitive, accurate sensors for that. Not only for developing for that and current medical service. Let me explain one thing for the coronavirus testing it here. So they first to get the test, for example, blood sample or saliva and that kind of thing, they needed to extract and separate out. It takes more than hours. And then they needed to amplify, we called the PCR. And that is another more than hours. And then they run the electrophoresis and they also detect the more than 1 hour. So that's why we need about 6 hours to finish up to see your result. And that is the old way. But in nowadays, we can just drop your specimen about less than 10 microliters, and they will go to the separation and then go to directly detect right away. So that is what we want to do it. So then currently, we often use the real-time PCR, which is a little bit longer time than what we wanted to do it. But however, at this moment, this is I think it's the high-tech technology they think currently, but we are more likely to consider in the future prospect, as you can see this slide. And this is the one example of the sample preparation and detection of this. If you drop the blood, and then we can separate out your plasma very simply by this simple flow rate. And then we can detect the whatever DNA or RNA or any biomarkers based on this technology. And this kind of thing we call the microfluidic channels. And this will be very powerful to separate out. Then today I would like to show you how we can prepare this kind of microfluidic channels by inexpensive method with a high throughput methodology, the technique. So that's one of we call the large scale additive manufacturing method. And this microfluidic channels, basically we can separate out whatever cells or white blood cell and this kind of thing based on well-designed, how the fluid channels. I think I cannot cover this during this class because you may consider the fluid dynamics about this on the micro channels, how you design of this on that kind of thing. And for example, you will see this, and if you get some small urine samples and then we put it here, separate out, and then we can detect whatever cancers you have from there, okay? The efficiency is pretty high. And even so far, this is the current technology. We still have a space. Can we make more simple and more faster than previous. Okay, so that is what I want to show you this. And the other thing is, this is the perfusion channel array. You can see more than 100 arrays, you will get this one. And each well has the very small cell, which is the stem cell grown, based on stem cell grown about the organoid. So each organoid will grow on each one. And then we can detect how the cell activity and physiological property, and then we can monitoring by this each perfusion channel as shown in these structures. Okay. And so the innovative way to making it is like a roll-to-roll manufacturing. So this is like printing the magazine or printing the newspapers. Can we do that like a printing method to produce on this kind of perfusion channels and also this kind of the diagnostic kit for this? Okay, so that is what we called the large-scale additive manufacturing platform, we call it. And this is the concept image. As you can see and hear, the printing unit, I think it's even India has a very good printers. It's like Gravure, Offset, and Flexo, and that kind of printers. If you slightly modified that, and then you can make it to print electronic device and integrate with Biochip. So this. Okay, so it's depending on how you designed the printing unit. For example, I show in here A to Z in here, okay? And depending on how you integrate, you can make the flexible diagnostic kit, which is the point of care, we call the POT. And that kind of thing is you can integrate microfluidic channels and sensor array, and even you can contain the batteries and wireless communication module in here. And if you just to simplify the AIJ, and then you make some water diagnostic kit, which is to diagnose the water quality and simple your drinking water, so whatever that kind of thing we can diagnose very quickly. But if you integrate the older line, including the even nano printing, and then you will get the arrays of the organ on a system. So in here, about the 400 the micro well in here, and each well contained your brain cell and your lung cell and your some liver cell, and that kind of thing is contained here. And then we can monitoring it to depending on how you treat it or your drugs. And that will be pretty much personalized on you. And that information will go through your smartphone and will go to the big data. And big data have analyzed your data and then give you back about your condition, or your data will transfer to hospital or your personalized doctors and can see what's your problem in your in this testing and here. Okay, this is what we wanted to do it. So especially this printing unit, as you see, we may printing more than 50 layers. We needed to consider each layer overlay printing registration accuracy on this. Okay, so that we can integrate the many devices on a time. Okay, so I'd like to show you how we do this in here in the lab. Okay, this is the first printing unit, and then you'll see this. And when we do this first printing unit, and you need to maintain the web tension very well. Okay, so you need to control the web tension, which is the non-linear function, so that you need to well optimize the maintained each printing unit, and then you need to get the web tension controlled very well. And second important issue when you do it in here, and then you will control the temperatures, which is the drying chambers, and then ink transfers from the Gravure roll in here. Okay, this is Gravure printers, and then you will see the ink rheological property. You need to understand how ink shear rate is changed, and then change the viscosity, and that will change the ink transfer and that kind of thing. Okay, so that we can control the ink patterns and that kind of thing system on it. And when you go there, and then second printing unit, and then the second printing unit, you need to control the registration of overlay. Okay, and of course, then you need to control the curing, the drying, and we need to understand how your printed layers is drying pretty well, and what kind of behavior you can get it by the simulation of your drying behavior of your employed the ink. And then we need to also control, as I mentioned before, the registration accuracy. And the rich registration accuracy, we need to control about plus minus 20 micrometers at here. And while we are controlled, as you can see in this data, about when we printing about 100 meters, we can control the registration accuracy about plus minus 20 micrometers. And based on that, we will keep printing it to electronic device and that kind of thing. And then all this system is controlled by the simple softwares. And as you can see, but we need to optimize this, the artificial intelligence, which is maintained by the machine learning system. So we designed the patterns, and then we needed to align, we needed to extract all the data which we obtained by this roll-to-roll printing process. In situ, we are optimizing this. Okay, then this is the system, you can see it. And finally, this is the imprinting method. So finally, you will get the microfluidic channel and this kind of thing. So I will show you this on here. This is the little bit locked in here, but we can imprinting the microfluidic channel. I will show you next slide about this a little bit more clear one. So you can see this is optimized a little bit, and this one is the microfluidic channel, perfusion channels, which is to control and separate your blood and your specimen. So that is we are making this microfluidic channel by roll-to-roll. We call the imprinting method. And what my student is spreading it here, UV curable resin, and this is the final step. And then this is the resin is cured by the UV light, and then the pattern is imprinted by the pressure on it, by the tension on it. And then this is I think it's the printing speed is about 0.6 meters per minute. Okay, so now you can see this is the high throughput. Actually, you can, if you follow this, and then you can manufacture actually to make the microfluidic channels or perfusion channels. It's about more than 10,000 samples per day on this kind of system on here. You can see, and then also the result is very consistent in here. Okay. So this method will be very powerful to prepare the biochips using all printing systems. So we are not etching away anything. So we just editing. So we don't have any waste from here, any side product and byproduct, which will contaminate the environment. So this is like a future manufacturing process. So we don't need to give any side effect to the environment, which we are suffering nowadays. So this is the method we are doing. So if we are successful in the near futures, actually this is actually we can monitoring everything. Okay, it is we can monitoring your food, whether this food is contaminated by what is the pesticide or any poisoning, some chemicals, or it is imported from the China, or imported from Korea, or imported from the India, who made this, and that kind of thing we can see quickly. And then also we can detect the virus and also the bacteria and that kind of thing is easily. And even we can monitoring the waters and also the environment, and also for the we can monitoring the aging, like monitoring the brain cell on this kind of thing. And this is almost everything we can do it. So very quickly with the inexpensive method. So the cost of each diagnostic kit, we are targeting about less than $1. So that kind of thing we can achieve by due to this roll-to-roll manufacturing process only this. That is why the class is for large advanced manufacturing platform. That is the targeting for this. Okay, so this is the for the wrap up the final slide for these lectures. And thank you for your attention. And if you have any questions, any interesting to study in Korea, especially in my lab, and then you may contact me through this email address. So and then you may send me your CV and that kind of thing, and I will consider about it. Okay. So thank you for your attention. So I think I'm finished about 3 minutes earlier. So I think we can I can give you any I can give you answers whatever your questions. Thank you.
Good evening, everyone. I am humbled and fortunate to have given the opportunity to give a note of thanks this evening. It was indeed a great opportunity to listen to Dr. Cho. The delivery was extraordinary and the concepts were explained in a clear way. I would like to express my sincere gratitude to the speaker, Dr. Cho, for being generous in accepting our request to be a part of the Professor series. We are very thankful to our management, respected GM Development, Dr. Jaikma sir, and our beloved principal, Dr. V. Velmurugan sir. My sincere thanks to Dr. Joe Patrick sir, head of the department, Dr. Zeenath ma'am, Professor Ebenezer, and all my colleagues and student volunteers for the support. This webinar has been made successful only because of the well-wishers, followers, and the participants of this event. On behalf of our institution and management, I sincerely extend my thanks for all your support, and I request you to keep supporting us in all our activities in the future. Thank you, everyone.